larval diapause rates. The results show that 8 to 21% eggs laid by female emerging on the first day of the short 4-week flight time would produce larvae that reach diapause. Females emerging 7 days later produced only 1 to 5% of larvae that reach diapause. As suggested by Pratt and Emo, the larvae of some Nymphalidae lepidopteran families go into extended diapause in hot, dry years as an adaptation to low food resources. Despite 2013 and 2014 being dry drought years, there was a population of adult butterflies that emerged. This population of adult butterflies was relatively small and scattered, and therefore field observation time was short and limited. Studies show that nectar sources are selected on oviposition sites. Euphydryas editha has shown it can change the relationship between host preference, host encounter rate, and realized diet, and therefore influence preference. Adult food choice is based on the availability of host plants and location of the butterfly. The research gaps I found was, are adult butterfly nectar resources affected by nitrogen and grazing, and the interactive effect of nitrogen and grazing on abundance of early nectar resources. My project goals were to look at the effects of an increase in nitrogen and grazing on butterfly fecundity and fitness by mainly studying the adult food resource. Now moving on to my objectives and questions. My objective was to understand changes in nectar resources of the adult threatened bay checkerspot butterfly due to anthropogenic air pollution through understanding how increased atmospheric nitrogen might affect the adult butterfly nectar resources, and also understanding if grazing might change the impacts of nitrogen deposition on nectar resources. My research design mainly focused on if the growing season is very short and the species of interest are rare and patchy and the target invertebrate was threatened. Therefore, the study was designed to generate baseline data and to formulate questions and pave the way for future work. I was really interested in early season flower resource availability, as fecundity could be influenced by early nectar resources, and resources are limited in early season versus peak biomass season. The first question I asked was, does the adult bay checkerspot butterfly utilize a wide range of nectar resources availability on Coyote Ridge? How does nitrogen enhancement affect the presence of early spring nectar resources for the bay checkerspot butterfly? How does grazing affect the presence of nectar resources in the plots? And how does nitrogen enhancement and grazing affect abundance of early season native and nectar resources? My study site was Coyote Ridge in South San Jose, which runs adjacent to Highway 101. It covers about 2,800 hectares of serpentine outcrop. Serpentine grasslands support many native animal, plants, forbs, and grass species. The timing and quantity of rainfall is highly variable from year to year, which significantly influences species composition. The map shows all the potential serpentine sites for the bay checkerspot butterfly in the Bay Area in red. Coyote Ridge, although, is the last known refuge for recurring bay checkerspot butterfly populations. This pie chart was taken from the Santa Clara Valley Habitat Plan. It shows the type of deposition in the vicinity of bay checkerspot butterfly habitats. Deposition is mainly from dry deposition from anthropogenic auto emissions. This atmospheric deposition accumulates and settles as fog in Coyote Ridge. Due to this increase in this atmospheric nitrogen, grazing has been implemented as a management tool since the early 60s. The current grazing regime at Coyote Ridge is 1 cow-calf pair per 10 acres for 6 months. Serpentine soil again is low in nutrients such as nitrogen, high in heavy metals, and several plant species have adapted to these conditions. This grassland generally has annual fluctuations in the abundance of plant species. Due to the unique abiotic conditions, Coyote Ridge hosts many rare endemic species of annuals, perennials, bulbs, flowers, forbs, and grasses. These species are typically slow-growing stress tolerators rather than fast-growing dominance. For my study, I utilized a two-factorial experiment with two levels, nitrogen addition and grazing, as the two factors at 10 locations, each within a few hundred meters at Coyote Ridge. These plots were set up by Dr. Jake Basari and Dr. Dan Hernandez in 2007. At each location, there were two 5 by 5 meter split blocks. One block was fenced off with barbed wire to exclude grazing by large mammals, mainly cows. Each split block was then divided into two halves, and one side was enhanced with nitrogen through fertilizer, and the other represented ambient nitrogen. This yielded a total of 40 plots. All plots were set up on different topographies, and data loggers were placed to monitor differences in temperature and humidity. Each block had a vegetation bar marked to ensure consistent placement of the quadrat on each sampling day. Data collection had two components: qualitative observation of what nectar resources the adult bay checkerspot butterfly was potentially using, collecting presence and absence data, and the quantitative portion consisted of collecting abundance, a percent cover of vegetation. Initial data collection started out in 2014 to map out plots and identify plants. To capture early season flower growth, plots were scanned twice a week for presence or absence of vegetation growth. On February 21st, the first set of qualitative data was collected for presence of early flowering resources. This was repeated on March 18th and March 21st. The quadrat was further divided into nested quadrats to accurately measure percent cover of each present plant in plots 1 through 5, 9, and 10. Percent cover data was collected on March 23rd, 27th, and April 10th. Upon emergence, adult bay checkerspot butterfly activity was observed around plots for a total of 6 hours. For question 2 and question 3, I used a chi-square test to analyze the qualitative data, as it would explain statistically if the observed results were a chance or if there was a real effect of the treatments. The null hypothesis for question 2 was the proportion of nitrogen-enhanced plots that contain nectar resource species used by the bay checkerspot butterfly will not differ from the proportion of ambient nitrogen plots containing such nectar plant species. Null hypothesis for question 3 was the proportion of grazed plots that contain nectar resources species used by the bay checkerspot butterfly will not differ from the proportion of ungrazed plots containing such plant species. For question 4, the null hypothesis was percent cover of native vegetation in the nitrogen-enhanced plots will not differ between the grazed and the ungrazed plots. I used the general linear model which can be used to determine whether means of two groups differ. I calculated ANOVA values using a least square regression approach to describe the statistical relationship between treatments and percent cover. Now moving on to my results. For question 1, does the adult bay checkerspot butterfly utilize a wide range of nectar resources available on Coyote Ridge? Adult butterfly foraging behavior was observed upon emergence for 1.5 hours on the first 2 days and 1 hour each for the next 3 observations. The available nectar food sources mainly included mid to late blooming flowers. Out of the available nectar sources, the bay checkerspot butterfly utilized 50% of the available sources on the first and second dates and about 60 to 75% on the last 2 dates. These observations showed that the adult almost utilized all available nectar resources available. Research question 2, how does the nitrogen enhancement affect the presence of early nectar resources for the bay checkerspot butterfly? Bloom times of flowers were recorded for growth in each treatment. Early season was defined as February through March 18. Mid-season or peak biomass season was mid-March through early April, and early season was defined as early through mid-May. Miola maritima is an early season flower as seen by its first flowering date, and allium is a rare plant that blooms later in the season. This graph shows the presence and absence of the perennial bulb allium in enhanced nitrogen plots versus ambient nitrogen plots. There were 20 plots because at each location, all the nitrogen treatments were clumped together. The graph shows there were more nitrogen-enhanced plots with allium sera with the blooming with a p-value of 0.072. This was an unexpected trend, but this trend was observed on one sampling date, and there was a difference of only 3 nitrogen-enhanced plots with allium. In contrast, Miola maritima was present in more ambient nitrogen plots versus enhanced nitrogen plots with a p-value of 0.091. As expected, Miola maritima tended to grow in fewer plots that had nitrogen enhancement. Question 3. How does grazing affect nectar resource plants present in these plots? Again, the grazing plots were clumped together that produced many plots. The graph shows the presence and absence of the perennial bulb Chloragulum pomeranianum. There were more ungrazed plots that had Chloragulum present compared to the grazed plots with the p-value of 0.077. Again, this trend was not expected. Question 4. How does nitrogen enhancement and grazing affect abundance of native and nectar resources? This graph shows the cross-treatment interaction on early flower resource with a p-value of 0.096. This was again a trend, and it showed that the enhanced nitrogen did lower abundance of early nectar resource in both ungrazed and grazed plots, but there was no difference between the ungrazed or grazed. The ungrazed ambient nitrogen cross-treatment plots had the highest overall percent cover of early nectar resources. The cross-treatment interaction of the nitrogen enhancement and grazing for native vegetation had a p-value of 0.061. The nitrogen enhancement tended to reduce native vegetation in the grazed plots. This showed the nitrogen enhancement tended to decrease percent cover even in grazed plots. Grazing was not able to lower the effects of nitrogen, while it is implemented as a management tool. So summary of results showed that the adult bay checkerspot utilized almost all available nectar resources. Nitrogen enhancement tended to hinder plant growth as seen by Miola maritima. There were high number of ungrazed plots with Chloragulum. And the nitrogen enhancement tended to reduce native vegetation in the grazed plots. Then enhanced nitrogen also lowered abundance of early nectar resource in both grazed and ungrazed plots, but there was no difference. Moving on to my discussion. The two main characteristics of serpentine is spatially complex landscape with low patchy productivity. My data collection was limited due to high variability. Rareness of these plants also made it more difficult to find statistical significant differences. My results showed that nitrogen did hinder plant growth, as seen by Miola maritima, which was present in fewer nitrogen-enhanced plots. If nitrogen is affecting adult bay checkerspot butterfly, which this could eventually reduce fecundity. Also, grazing did not seem to mitigate the effects of nitrogen deposition on native vegetation growth. Thus, in absence of a policy change to reduce nitrogen emissions, a re-evaluation of the most effective means of controlling exotic species in the serpentine grassland might be needed. Action Pesari studied the interactive impacts of grazing and nitrogen on species richness during peak biomass. They support the use of grazing as an effective management tool, but also suggest that grazing may become less effective as nitrogen accumulates through atmospheric deposition over time. Also, more targeted grazing management might improve conservation outcomes at this site. If grazing may reduce adult food resource, then we need to be careful about recommending grazing as a management tool. As shown by my results, nitrogen did seem to increase native vegetation growth in ungrazed plots, contrary to other studies at Coyote Ridge. To confirm this, sampling more plots and gathering more data points might be important. Although nitrogen cannot be removed from an ecosystem, local management can implement strategies to implement new conservation projects in the future focusing on these impacts. Targeted management may be required to improve conservation outcomes, evaluating the full scope of ecological effects. Also, Coyote Ridge has been experiencing nitrogen deposition for several years, and the amount of simulated nitrogen may not be an exact measure of the magnitude of its impacts. Studies to study current rates and the outcomes may vary in pristine serpentine grasslands versus Coyote Ridge. Continued studies to understand adult nectar resources to better understand butterfly ecology might need to be continued. Increase in urbanization combined with nitrogen emissions is pushing the threatened bay checkerspot butterfly and serpentine ecosystem to extinction. Monitoring and better understanding the widespread impacts are important to help protect these biodiversity hotspots. And that brings me to the end of my presentation. Any questions?
We have time for questions. People have questions from the generic audience? We'll turn to the committee. I wonder if you could go back to your slide of the late season flowers, Clarigalum or the this one here, or then you also had Allium.
Yes.
That's also late season, right? So explain a little bit more. So these are your unexpected results.
Yes.
All right. And I think there's always a temptation to sort of say, so we'll just move on. But what does it mean when the native plants, some native plants are reacting really differently than other native plants? So you've got a plant that is reacting positively to pollution.
Yes.
And it's potentially a late season resource for an endangered butterfly. So
yeah,
just thinking about that as a potential problem. Now I know that your p-value isn't definitive, and but if it were true, particularly in terms of grazing, which we have more control over, the one where you had the Clarigalum had a negative response to grazing, what would it mean? What could we do about that?
Yeah, this trend was unexpected, especially because it's a bulb, and Neola is also a bulb, which showed completely opposite effects. But it could mean that grazing could be an effective management tool, but maybe for late season to maybe help these late season flowers.
So in this case, what's happening?
The allium there were more plots that were enhanced with nitrogen having allium blooming in them. So yeah, it did seem like the nitrogen was helping these. We would have to see what the effects of grazing would be on this.
But assuming just this alone, it's one of those things that sometimes there are some individual plants or some individual resources that will deal with humans, and deal with humans in a way that their comrades don't. And so, in a sense, I find myself wondering, in some climate future, when late season is important for the butterfly, is allium going to be more important? Or is this such a it's very, very rare, it's just not enough for
it? It is very rare, and yeah, late season flowers would be important to the butterfly, because if we look at this threatened butterfly, it has really small populations. And the very first ones and the very end ones would be the most important. So whatever is blooming in those seasons could potentially affect their.
And the Clarigalum is even more interesting. Go forward to that one.
Well, before you leave this slide, these combine grazed and ungrazed, right? That's why your N is 20. This is a combination of grazed and ungrazed.
Yeah.
So you're kind of conflating that in these results. And I think, that might be, part of the, if grazing is an important factor, that's not being separated out here. So I think the fact that the N is 20 is a little, I don't know, maybe a little deceptive, I guess,